Hi, Jared. How are you doing?
Hello?
Hello. Hello. Can you hear me?
Hello?
Can you hear me? Hello.
Hmm.
Hello.
Oh, yep, I can hear you.
Okay. Awesome.
How are you today?
I'm doing good. How are you?
Oh, living the dream, right?
Yeah, that's true.
It's it's Friday. It's got a good presentation coming, and then we're all gonna enjoy the weekend. It'll be good.
I hope the weekend remains like this.
Oh
Hey,
yeah.
Thomas. How are you?
Oh yeah. It'll be good. Get outside, take it easy. It'll be nice.
Can everybody see my screen?
Yes.
Awesome. I
Yep,
see
can see you.
it's already noon, and we have all the participants as well as our judges. So I'll get started. Before we start, I have a small script that I need to read through. So to give an introduction about this, about how the event would be and how we plan on continuing. So first of all, thank you all for coming out and taking part in this year's Graduate Research Colloquium. My name is Ronit Karmakar, and I will be your moderator today. Just a couple of housekeeping rules before we get started. Each contestant will have about 12 minutes to present. Your time will start the second you start speaking. I will give you a 1 minute warning by interrupting your talk. I'll make a noise like this. Was that audible? Awesome. Once you're ready to present, give me a verbal cue like start, etc., and I'll advance through your slide deck. During the presentation, give me a verbal cue like next, advance, proceed, next slide. To advance your next slide, I will introduce you before you talk. You can jump directly into the presentation to save some time. And at the end of the presentation, judges can unmute themselves and ask questions to the participants for about 5 minutes. While the judges are scoring, I will moderate questions from the general audience. I ask the audience to stay muted during the presentation and drop their question in the chat box during or after the presentation. And to limit the distraction, I would request everyone else to keep their um, video off as well, or while someone else is presenting. So maybe if the judges and the presenter keep their video on, I think that would be best. And with that, I would like to welcome our first presenter. Does anyone have any question before we start? All right, seeing no hands, I will first welcome Manas Warke from Biological Sciences, and he would be presenting on developing a model to understand the effect of soil arsenic on human skin cells. With that, Manas, the floor is all yours.
Thank you, Ranit. So just a few things. In some of my slides, there's one image on the left side. So in those slides, just press next twice so that the entire image deck is there on the slide.
Awesome. I'll try to keep that in mind in case I miss. Just say next one more time.
All right, sounds good. Thank you. So hey everyone, thank you for joining in. So in this presentation, after like initial background on how arsenic contamination takes place in soil, We'll look at how we are looking to develop a model to study the effect of soil arsenic on human cells using cell culture. Next. So, arsenic occurs naturally in the Earth's crust. It is present in the air, water, and soil around us. In nature, it occurs in the form of sulfide and iron ores. And it was discovered in 1200s, and since then, we have exploited the use of arsenic in many ways, and it is used in many industries in today's world. Today's world. Arsenic is also very lethal to human health, and it causes cancers of skin, lung, kidneys, and prostate. It is also linked to the diabetes, obesity, and various skin and heart diseases. Next slide. So, realizing the effect of the harmful effects of arsenic to human health, it was classified as a class 1 carcinogen by major environmental and health organizations in the late 1980s. Next slide. And can you press next once more? Yep, thank you. So looking at the world map for arsenic pollution, you can see all the shaded regions that arsenic pollution is like widespread all over the world. And these markers are basically considered as arsenic hotspots. And these locations are also like agricultural fields, which shows that the arsenic pollution in soil is mainly concentrated towards the agricultural fields. Next slide. So, what causes arsenic? What causes arsenic contamination in soil? So, there are two major reasons. The first reason is natural, combined with human activities. The first example for that is using arsenic groundwater for irrigation. So, in many regions around the world, especially the arsenic hotspots, which we saw in the previous slide, there is arsenic contaminated groundwater present, and the overuse of this contaminated groundwater for irrigation has led to like the spread of arsenic in the soil and the land around it, leading to more arsenic in the agricultural fields. The second reason is mining. So in naturally contaminated sites where there is arsenic present already, moving the landmass and using heavy machinery in that area leads to like spread and accumulation of arsenic in the surrounding area. Next. Another reason is human induced, that is the extensive use of arsenical pesticides for maintaining and improving the crop yield in the agricultural fields. And the other reason is using copper chromated arsenate to maintain the quality of logs to make wooden decks and etc. So both these applications were banned in 2010 because until that point we knew the harmful effects of arsenic, but we did not know what happens due to the chronic exposure of arsenic. That is low arsenic exposed for a very long period of time. Next slide. And the main reason for arsenic contamination in the US is just widespread use of pesticides in the crops and the groundwater with the sole aim of improving the crop yield and the quality of crops in the fields. There are at least three case studies which have reported extensive use of arsenic, like the cotton fields in Texas, apple orchards in New Jersey, and potato fields in Idaho. And these are just some images that show the extensive use of pesticides in the fields. Next slide. So, from all of this, we should consider that arsenic exposure from the soil is a major concern. Arsenic exposure takes place through three major pathways: ingestion, inhalation of arsenic dust, and through direct skin contact. The main reason for this exposure is the hand to mouth activity, and we need a model to test how soil arsenic can affect the human health. Next slide. And a study has shown that if we use arsenical pesticides for only 3 years, it leads to an accumulation of 900 milligrams per kilograms of arsenic. Imagine the amount of arsenic being accumulated in the fields due to the continuous use over the period of decades until 2010. Next slide. So, can you press next once more? Yeah, thank you. So the another reason is that most like only the European Union recognizes this concern and has placed the safety limit of 20 milligrams per kilogram of arsenic in the soil. But other major health organizations like the WHO, FAO are still to recognize this effect or the concern. And the major reason because this is not a safety limit due to that there's no model or a protocol that could test the effect of arsenic on humans' health. Next slide. So there are models that study the effect of arsenic, but they look into the arsenic accumulation in the blood, urine, and toenails, which are not very effective biomarkers because if you are looking at arsenic in the toenails, it has the arsenic has already crossed the blood barrier, and we need to look into more cellular and molecular mechanisms to see what is happening. And if we use animal models to study that concept, it will be very expensive and time consuming. So we propose using cell culture techniques to study the effect of soil arsenic at a cellular level. Next slide. So we proposed, first started with the tea bag model. The idea behind this model was to use a tea bag with soil and then put it in the media for the cells, and then hope that the arsenic moves from the soil to the media, and then we can treat the contaminated media on the cells. Next slide. So we started with incubation of the arsenic treated soils in the greenhouse, filled them in the tea bags, and the tea bag was transferred to a beaker containing media on a shaker overnight. And next slide. And once more, next. Yeah. So in this, so we got arsenic in the media, which was good. But there was a lot of arsenic being trapped in the tea bags. And so we had to reject this model because we thought that the tea bag would act as a filter and not as an absorbent for arsenic. So we rejected this model, and we moved on to the fractionation model. Next slide. So in this model, we are just looking at targeting the bioavailable fractions of arsenic in the soil and using those to treat them on the cells. Next slide. So and next two more times. Thank you. So how we do that? So what is bioaccessible fraction in the soil? So in the soil, arsenic exists in these six different forms. Not all of them are readily uptaken by the human body. For example, F1, which is the water soluble fraction, can be easily absorbed by the cells and water and can be taken up by the human cells, while F5 and F6 are strongly bound and cannot be taken up by the human cells. So the idea behind this is to extract the water soluble fraction and then test it on arsenic. So next slide. So in this model, we just start with preparing the soil in the greenhouse. Next. And then we use the standard protocol for extracting water soluble arsenic using 1 gram of soil per 15 ml of water. Shake it at room temperature for 2 hours. And next. And remember that this is just water in the shaker. So we used concentrated media and diluted it with the arsenic leached water, filtered it, and the media was ready for treatment. Next. And once more, next. Yeah. So comparing the two models, we favored the fractionation model because there was no arsenic being trapped in the tea bags or any other medium. We targeted the bioaccessible fraction, which was more a logical approach, and there was no contamination in the media, and the cells grew just fine. Next. So after having enough evidence that the model is working fine, we tried to look at the skin exposure by simulating what happens if we treat the soil arsenic on skin cells. We use skin cells first because it is the first organ that is exposed to arsenic in a contaminated site. And the soil concentrations were based on the 10-year accumulation of arsenic due to pesticides. And we use this Hecat cell line because it is an established model for testing arsenic. Next. So first we looked at the cell viability and how what happens to the cell and the effect of the arsenic due to 5-day exposure. The negative control in this experiment was just pure media without any soil or arsenic. Control in the experiment was soil but with no arsenic, and the respective concentrations of 45 to 25, 450 and 900 milligrams per kilograms. If we look at the second graph on the right, and can you press next once? Yeah. So as the arsenic concentration increased, the cell viability decreased, which was according to our hypothesis. But at the very highest concentration of arsenic, we saw that the cells managed to grow even like for a period of 5 days. So we wanted to see what was happening in the cells, and we did the scratch wound assay for that. Next. So basically, in the scratch wound assay, we have a confluent well of cells, and then we make a scratch, so that basically making a wound on the cells, and then observing the cell growth and migration over a period of time to see how the cells are recovering. Next. So for this slide, please increase the brightness so you can see the images clearly. And so in this, can you press next once more? Yeah. So in this, again, we use the same concentrations of treatments, and after the wound was made on day one, the cells continued to recover and migrate. At day 3, we observed that the negative control and control cells grew just fine. And as the arsenic concentration increased until 450 ppm, the cells were not were dying. Like they were not able to recover as good as the control cells. But at 900 ppm, as we saw in cell viability, the cells were continued to grow and there was not major change in the cells seen. The cells remain attached, they were living, and they were kind of growing as well. So next. So based on this study, we concluded that our model is effective, like looking at the bioavailable fractions from the soil, extracting them and then treating on the cells. So after these two experiments, we have already looked into like expression of cancer markers using immunostaining. And then we also hope to like simulate the similar conditions of ingestion and inhalation pathways using intestinal cells and uh, epithelial, bronchial and tracheal epithelial cells. So the bioavailable fractions for these two will be different. So the protocol changes for the whatever are extracting the arsenic, but the idea remains the same to extract the fractions from the soil and then testing them on the respective cell lines. Next slide. So that's it. Thank you.
Thank you so much, Manas. Excellent presentation. Now request the judges to ask their questions. You'll have 5 minutes to ask them.
Okay, I'm Sang-Yun Han in biomedical engineering. Yeah, so it was good talk. I learned a lot about this arsenic and your experiments. Yeah, so maybe you did the two experiment, one with the viability, the other with the wound healing assay. You showed that with the arsenic, the soil arsenic containing soil kind of concentration, more concentration, or kind of any concentration, there's no ability for cells to close the wound. So, and then this wound closing assay or wound assay is representing both proliferation of cells and also migration of the cells. What part do you think this arsenic affect the cell's ability? Like proliferation or migration or both? And if do you have any kind of like potential pathways or mechanisms how arsenic can affect those abilities?
So yeah, so arsenic basically, due to from the research that we have seen, arsenic affects both the cell proliferation and migration, but at certain concentrations, the cells are take up the enough arsenic that they can be converted into cancer cells. One such pathway is the GST pathway, glutathione pathway, where glutathione kinds of binds with arsenic, and then all the checkpoints are altered, and then the cells become cancerous. So that is why we are looking at the cancer markers like CD44 and the EMT proteins like cadherins and ZEB1 to see what is happening at those higher concentrations, if the cell are dying or they are just converting themselves into cancer cells.
So if let's say cells are converting to cancer cells, are cancer cells not migratory, not proliferating? People believe that they migrate more, they also proliferate more, right? So what causes this stoppage of this closing? That would be a little bit different than the cells transforming to cancer, maybe.
Yes, so this is just the preliminary studies. We are just, and the 3 days, we just did the scratch wound for 3 days, and I think that is a very low time to determine if they are going to convert themselves fully into cancer cells. So hopefully by after the preliminary state of data, we can just increase the time length of the experiment and then see what is actually happening. But from the initial experiment, we saw that there are some expression of those cancer markers seen in the higher concentrations. So maybe a future experiment we can add those definitely to see increase the time length and then see if they're like recovering at a higher concentration again.
Yeah, and also the migration or proliferation, there's like key molecules there like that governs that proliferation or migration, like GTPases, actin polymerization, things like that. Maybe you can also look up those pathways too.
Yes,
Yep.
definitely. So the idea behind starting this model is to like just replicate if we can use this model to see what is out in the literature and then get the same similar results to conclude that even soil arsenic is also as harmful as arsenic in water, and that
Mm,
our model can be used like as a model to treat any contaminated soil on human cells and then use it for future studies.
right. Okay.
Mhm.
Thank you.
Thank you.
Can I still ask a question, or are we done with the judges' time?
No, you have more than a minute now.
Okay.
You can ask.
I was just wondering if you could just elaborate a little bit on the European. You mentioned the European Union had different standards. Like, is it backed up by certain research? Like, how does your research place into that?
So the standards are basically set up based on the background research done at various like based on the research, and the European Union is the only one who has recognized that soil and arsenic is a major concern. Uh, Probably the other institutions are just waiting for more research and more data to conclude that okay, we can set this as a standard limit, because 20 milligrams per kilogram is quite high, I guess, for like uh, as a safety limit. So it should be lower, as my personal opinion, but maybe just the organizations are just waiting for more data. And again, this model would like show that how much arsenic is actually harmful for us.
Thank you.
Mhm.
Right. Thank you so much, judges. Thank you, Manas. I would like, while the judges are now scoring the contestant, I would like to ask if there are any questions from the audience. If there are any questions, feel free to post them in the chat, or you can unmute yourself and ask directly. Right, I'll just wait 20 more seconds to see if there are any questions. Otherwise, I'll just move to the next participant. Seeing none, thank you so much, Manas, and I wish you all the best.
All right, thank you, Ranit.
We have our next presenter, but before I move there, judges, are you done with this scoring? Should we move to the next presenter?
I'm just done. I don't know what the other judge.
Dr. Jacob.
I'm almost there, but
Okay.
yeah, I think we can roll through.
Right. So we have our next presenter here, Jared Edwards from the Department of Chemistry, and his topic of presentation today is discovery of Hemolysin X, a bioactive compound with therapeutic potential. With that, Jared, the floor is yours.
All right. Hi, everybody. I hope you're all having a great day. As I said, I'm Jared, and I do research under Dr. Dom. And today I'm going to be talking to you about Hemolysin X. And the first thing really that comes to mind is what the heck is Hemolysin X? So with that, you can go next. And so Hemolysin X is a plant-based biomolecule, and it's capable of lysing multiple cell types. And what that actually means is that with these target cells that you have, you're able to expose it to helix. And upon that exposure, you will see rupturing of the cell's membranes, where holes will be kind of punched into it, and it results in the target cell's death. So as a result of that, you're able to have a large amount of lytic activity, and you're able to completely vanquish these cells. So this is a pretty interesting thing. And what really makes it useful in terms of detecting and all that is, if you can go next, please, is that it's very functioning towards red blood cells. So that's where the hemolytic action comes from. So what you can see here in this slide is what intact red blood cells look like in these tests. You see they're forming a nice button in the bottom of the wells of the cell. And that's nice, healthy cells. They're just in PBS, they're fine. When they're treated with this Hemolysin X, they take on this glassy appearance that you see on the bottom there. And what that means is the cells are completely dead, they're lysed, they're ruptured, all their contents are released. And it's very useful and effective, and it displays how voracious this molecule really is. And it's very, very useful when you consider how many extractions we've done with this plant-based molecule. If you can go next, please. We can see here that we have used PBS, methanol, ethanol, et cetera, all these different solvents to extract this plant material. And in all of them, we're yielding this helix molecule. We're witnessing this hemolytic activity that we've seen in the previous slide. What makes this really wild is that if you put two of these extractions, so if you were to take isobutanol and ethanol, and if you were to extract in one and then put it in a set funnel and put it together with the other one, you'll see a distribution of hemolytic activity into both of the solvents. So it has preferential within these extractions while also going into all of them and still displaying this activity. So, and you can even dry up the sample and then reconstitute it in a new solvent and it'll be active again. So it's a very amphipathic molecule. It's showing no true preference towards polar or non-polar. It can go into all these, and it's likely a very strong mixture of similar molecules overall. So what we really need to do from there is to work on purification of this complex mixture. If you can go next, please. So we see here, this is what a lot of my core research has been lately, has been high performance liquid chromatography. So HPLC of this hemolytic extraction. So we have many different fractions, many different extractions that are done. So it's just really a testament of just going through, performing HPLC on all these and seeing how we can spread this out. And what we see, if I adjust the gradient accordingly, I can get it all to elute out in one peak. It makes it look like there's a nice, so it just got one sample or it's just one core group of stuff. But if I actually stretch out this gradient accordingly, I see that I get many, many 20, 30 plus small peaks, as you see on the bottom down there. And all of these have associated structures and et cetera. They're actual samples, even though they look small, they are comprised of various compounds and species. And it's a matter of going through and identifying each of these and then trying to determine which ones are actually contributing to the activity of the sample. If you could go next, please. So to really kind of get a better idea of what's in each of these, I've been performing MALD on the received samples. So matrix assisted laser desorption ionization is basically a quick and easy mass spec analysis that I can do of my samples. And what we see over on the A side there is what it looks like before HPLC purification. So this would be a more crude sample. This would be ran on a gel column through size exclusion chromatography. And what you look at and you see in this fraction is many peaks. You're seeing kind of that characteristic cluster that we'll see on a lot of these samples, where you're seeing at 1400, 1500, 1700, 1800, et cetera, little group peak over an 890 and 1000 there. When you look at the actual HPLC fractions that you receive from that, you see denoted differences. So we are pretty confident that we are attaining purification of individual fractions and that we're getting different samples and we're not just getting some kind of mess. And you can see that kind of in the boxed in areas there with the kind of greenish red box there, where we're seeing the 997 peak getting surrounded and you're not seeing any of that 1000 peak appearing on the fraction 1. You're seeing the complete absence of that 2000 blocked in over in purple on the A over there. And we're seeing in fraction 2, you're seeing that reddish green box gone, but also you're seeing even in the individual peaks that you're kind of looking at it from afar, like, oh, that looks kind of similar. but those individual peak mass charge numbers that you're seeing are actually different too. You got 1414 versus 1411, 1560 versus 1576. And this kind of further, reiterates that point of, all right, this thing can extract in many different things, and it's likely got this mixture of very similar molecules that are coming out at different times. But if you push the gradient together, you also can get them all to come out at once, because they're not that different, but they're just a little bit different. So it's a matter of taking these things that are just, a fair few hairs breadth apart and kind of finding out which one is most important to this hemolytic activity. But we can say with some confidence that we are achieving a level of purification that we previously had it now. And now it's just a matter of going through each one of these fractions and further identifying them as necessary. So kind of moving on from this quantitative side of it, where we're getting some data in that, we're moving on to the qualitative, where I want to visually analyze how this hemolytic activity is happening and better gauge my understanding of it. If you could go next, please. So we're seeing here, this is the actual degradation of the cells upon exposure to varying concentrations of helix. So we see up in slide one, up there in the top left corner, what a healthy red blood cell is looking like under this field emission scanning electron microscopy. When we expose it to a low concentration of this helix, we are seeing that the cell becomes stressed. It is taking on the spherical shape as opposed to that discoid shape in the first one. As you increase the concentration further, you see further disruption of the cellular membrane, where you're seeing the kind of the rough circular portions appearing, projecting out of the cell surface at step 3. At step 4, you're seeing, when you're increasing concentration further, you're seeing a cell that's basically on the verge of death there, that is having all those pock marks and everything in it. That is a cell that is going to die. When you increase the concentration even further, you're seeing that in the 5th spot there, we're seeing a cell that is dead. It is dead. It's just had all these holes punched through the cell's membrane. And then on step 6, you can completely obliterate the cell if you have it at a high enough concentration, which, if I'm being honest, is not that high, most likely. You can achieve that result much easier than you can the lower ones. So you're having this very vicious activity approach by this helix molecule, and you're able to get it to stepwise basically points. So this could add a lot of value down the road, where if we find that it has certain affinities towards certain cell types, and that it has activity towards them, that you can actually tame it down and you can attain certain levels of varying levels of degradation. So it's valuable to get this information now on an easy model system that is the red blood cells. Furthermore, if you could go next, we are also visualizing in certain extractions, there's this pseudo hemagglutination activity that is also being present with the extractions. So what this means is that there's another molecule that may be from that same group of families that performs an activity that ultimately results in some lysis of the cells, but the way that it approaches it is definitively different. What we're seeing is our healthy red blood cell 1 getting stressed out very similarly to as the helix lysis molecule does. But as the concentration increases, you see these deformations of the cell's surface and its membrane. They kind of leach together and they start to clump up. And this gets more and more exaggerated as you increase the concentration further and further in 4, 5, and eventually the cells start to die in 6. So this is just a unique difference in between what some of these molecules are likely doing and how they achieve this lytic result. And it's just good to denote them now so that we can come back to it later when we further analyze each one of those peaks, and we can kind of identify, okay, maybe it's this cluster that's doing this type of lytic activity, and it's that type of cluster doing the previous lysis activity. And that way we can better understand how this is working as a whole, because this would be coming from, a whole sample that's not just purely doing this agglutinating and purely doing this lysis. In the crude, it would all be there present. You can work out a better understanding of how this molecule is actually used in nature, and then maybe how we could use it for our own biological purposes as well. Furthermore, to kind of cross-analyze this data, if you could go next. we also use atomic force microscopy to verify the results that we're getting in field emission scanning electron microscopy. So I just do this as a reassurance to myself and to basically test out the machine that is the AFM over there as well. And we can see that we can get this jointed images for these similarly treated cells, and we take them and we just use one in AFM and one in FESEM. We're able to get these replicated results. So it further kind of, amplifies that, okay, what I'm seeing is correct. What I'm seeing, I can believe in. This is verifiable, and I can repeat this. So moving forward to the next slide. We're trying to examine what we can do now and prepare for in the future. And so the main thing is really isolating each one of those active fractions through HPLC. So all those different extractions, all those different solvents that we use, and we want to continue to isolate those, get those individual peaks, analyze them in MALDI, and then reconstitute them in a more cell-friendly buffer, so like PBS, and then test them against the red blood cells and figure out if they are going to lyse at that point. And
I'm afraid
then
it's time.
All right, that's perfectly fine. Thank you very much.
Thank you so much for your presentation. Judges, the floor is all yours now for the questions.
If Dr. Jacob has a question, I'll let him go first. It's like taking turns.
Oh, sure.
Yeah.
I think that maybe my question might prepare the terrain for you. I'm just curious to see if you see your research going into cancer treatment research in that sense, if it's already been considered. And so, because I know you're in the chemistry department, right? But
yeah,
if you could just tell me a bit more about that.
Yeah, so we've done a little bit of investigation with that. We've seen cytotoxicity towards colon cancer cells, breast cancer cells, and some hepatic cancer cells as well. And that's been pretty preliminary though at this point. It's one of the things that we're really working towards figuring out further and analyzing more with these other cell lines. It was just done as kind of a prelim, just to be like, hey, can it do it? And our finding is yes, it does have activity and cytotoxicity towards these cells.
Yeah, so so following up with that, Dr. Jacob's question. So like so you're kind of studying this effect of this Hemolysin X, but maybe I missed it, but I kind of didn't get the why this is relevant, right? So like how this toxic material is affecting our life, or what is the implication? I know you are kind of going toward a little bit to the cancer treatment, but you didn't talk about the specificity of this drug or toxic material. So do you have any idea about the kind of this implication of this Hemolysin X? And
yeah,
also
sure.
you didn't define this HPLC. Can you define that too?
Yeah, for sure. So the big thing with Helix that makes it, like you say, interesting from just a toxic reagent to something that's actually usable is it does have specificity. So we're able to inhibit its activity through using lipid molecules. So you can use certain cholesterol, desmosterol, and you're able to inhibit its actual lytic activity using those. You can actually use pre-lysed cell membrane and use that to inhibit it as well. And it also, certain glycoproteins can inhibit it as well on top of that. So there is an ability to modulate its activity beyond that. HPLC is high performance liquid chromatography. So it's just basically you're going to be using a solvent system, and you're going to have a column. We use a reverse phase C18 column. And you're going to be injecting in your sample, and you're going to be adjusting a gradient of those solvents that you use. I use acetonitrile, and I use water with formic acid in it. And I use, you just basically modulate that gradient along till you find a system method that works best to actually elute off your sample in a manner that is separating it from each individual peaks. So that took a lot of time figuring out the specific method. I could go into more detail if you want, but
Mm-hmm.
yeah.
I see. And also SEM, the FESEM and the AFM, did you use the ECMO facility or?
Yep, I use ACMA all the time. Owen and Liz and all those guys over there have been a huge help through all of this. And the fact that FESCM machine is it's very good. Big fan.
Okay. So the environment is more kind of liquid. You cannot use a liquid environment. You have to evaporate them, right?
So basically, yes, you have to fix. So basically, what I'll be doing with that stepwise procedure, I'd be doing a normal titer test where I'd be using 96 well plate. I'd serially dilute my sample and have the red blood cells in there. And then midway through the actual titer test, the activity assay, I would be taking that sample out. I'd be fixating those blood cells, and then I'd be treating them accordingly, and then adhering them to a plate and all that. And then I would be preparing them for microscopy.
I see. Okay. Yep. Thank you so much. I'm done.
Yeah, thank you.
Thank you so much, Jared, and thank you, Dr. Jacob and Dr. Han, for your questions. Do we have any question from the audience now? So Thomas has a question, Jared, for you. If you can see the chat, or otherwise I can read the question for the audience. It's is Heli X consumed when it interacts with cells? Also, do the lower concentration that only damage the cells cause cell death after a longer time period?
Yeah, so that's actually really interesting. So I'll start with the second part. When you're using those lower concentrations, yes, actually, if you leave them exposed to the helix for an extended time. So if I were to, where I'd normally do these titer tests for like a half hour, if I were to extend it out to 2 to 3 hours, and if I noticed that there was activity occurring in those, but it wasn't full lysis, over time, eventually you'll see that those cells will eventually lyse. I actually did a test recently that it took a whole week for the next well to actually lyse, but I kept it refrigerated and concealed, and so it didn't dry up, and it actually over time eventually did pop it. So it is continually doing its activity as long as nothing's inhibiting it. If it's actually consumed, it's something that it's a little bit hard to do to determine. So there isn't a good way to really separate out the helix molecule from the membranous material if you're actually doing it at a high level. So it's hard to really like filter it out in any meaningful way. And also, if you try to do it by activity, it's hard to discern if it's just getting deactivated from too much dilution at that point, or if you're just eventually using it all up. So if like I've done tests where like I've taken a high active sample and I've just directly put it into red blood cells, and I've gone up to like 3 mils that it lysed all by itself, and it didn't really reach its capacity. So it's something that's kind of hard to discern because you can't really sieve it out properly after that point, but it may be.
Thank you, Jared. Thank you,
Yep.
Thomas, for your question. Do we have any more questions from the audience? Are the judges done with the scoring?
Not yet, maybe 1 more minute. Yeah.
Okay, sure. Right, while judges are
Thank you very much.
scoring, Jared, do you want to say something else that you want to mention? You have around a minute still left.
Nothing much. Just thanks everybody for coming. Thanks for listening. I appreciate it. As they were saying, use the Ackmal lab too. The Ackmal lab's good stuff. A lot of good microscopes, a lot of good stuff to use over there. Very useful. If anybody has any other questions, feel free to ask.
Dr. Han agrees with you.
Yeah, it's good stuff, man. I don't know. Those microscopes are fun. All right, I'll mute my mic. Thank you very much, everybody.
Thank you, Jared.
Thank you. I'm done. I don't
All
know
right.
about Dr. Jacob. Yeah.
All
Dr.
set,
Jacob,
Chair.
are you ready for the? All right, thank you. All right, with that, I would like to introduce our third speaker today, Thomas Ripplinger from Department of Chemistry, and his talk today will be on quantum mechanical investigation of gas absorption in MGMOF 74.1 and MGMOF 74.2. With that, Thomas, the floor is all yours.
All right, thank you. So as you mentioned, my topic is with MGMOF 74.1 and 2. I will just say MOF, and that means metal organic framework. So as I continue through the conversation, that's what MOF will stand for, is metal organic framework. Next slide. All right, so what are MOFs? Kind of three parts of the definition. They're periodic in 1, 2, or 3 dimensions. So they have repeating unit cells that can propagate in X, Y, or Z. They're microporous. So as you look to the right, you see a bunch of different structures, but you'll notice they all have one thing, is that they have pores built into their framework. So they're not just atoms right next to each other all the way through. They have some sort of gap where there's space for things to be or to pass through. And then the biggest part of the definition is that they're hybrid materials. So they're constructed of inorganic bricks connected by organic ligands. And because of the diversity of metals and the many different organic ligands that you could use to connect them, there's pretty much endless possibilities of frameworks. So what are these used for? So a few major topics that MOFs are investigated for is gas capture and storage, because of the metal sites, because of the ability to control the environment. They're very useful for being specific for a particular compound, or just for their binding of a large volume of structures. So you could have more than one gas per cavity, which is very beneficial in lots of applications for storage and capture. The tunability makes it very useful for separations, either in an open environment or in a controlled, if you just need to separate a compound that is not easy through traditional methods. There's a lot of investigation in their sensing capabilities because of the metals involved in these structures. They often have rich electronic fields associated with that can enhance spectroscopies, like SARS is sometimes used, surface enhanced Raman, or whatever else that higher sensitivities are needed for, as well as catalysis. Next. So into the naming convention, the beginning part is of the MOF is what metal site there is. So this case, in this study, it's all magnesium. So X would be the magnesium in this case. MOF 74 is the name of the structure type, and N is the number of benzene rings that connect the metal sites. So in the picture to the right, we see that there's only one benzene ring connected, connecting each of these metal vertices. So this would be MgMOF 74.1. What's special about this structure? So MOF 74 series have quite large cells. They're hexagonal, and they have open metal sites. So the magnesium, in this case, or whatever other metal you want to put in there, are not fully coordinated. There's an open spot for molecules to come in on and physiosorb, or in some cases, chemically absorb. Because you can adjust the size of the ligand, there's variable pore size, you can adjust that to your needs. You can have different metals. There's been attempts to use iron or nickel and to actually magnetize the structure by orienting the spins. You can also functionalize it, so you can substitute groups onto the metal or onto the ligand to give different properties. So the purpose of this work is to model the gas absorption in this structure, but particularly for the structure that's one size larger. So Mg MOF74-2. This is relatively unstudied, even though it has shown very promising properties. And then to combine the binding energies and the geometries with that larger MOF74-2 with MOF74-1, the one that's pictured here. And then to develop another method to study these using molecular clusters instead of the more computationally expensive periodic boundary conditions model. Next. So brief on the methods. We started off using the periodic boundary conditions model, or PBC, with different molecular loadings. For the PBC calculations, we use a plane wave basis set, had the cutoff at 35 Rydbergs. We used a PBE functional with a empirical, or not empirical, a non-empirical long-range interaction correction. And then depending on the method, we either froze the lattice parameters or relaxed them. For the cluster model, which we built, we used a 6311G basis set, a B3 lip functional with a D3 correction by Grima, and we kept the cluster frozen as we relaxed the gas onto it because it is not a full molecular structure. Next. All right, so results for geometry and energies. I just want to point out right away that if you look at the binding angle, the binding distances between each of the models, for the most part, all the gases absorb onto the metal site in very similar ways when comparing MOF74-2 and MOF1. The biggest exceptions for this are with ethane. So if you look near the bottom of the table, you'll see that there's different binding angles and binding distances between MOF74-1 and 2. And most notably, that the cell actually shrunk a good portion, actually 23%, when in the presence of ethane, and we relaxed the cell parameters. Another thing I want to point out is that in general, MOF74-2 absorbed the gases more strongly than MOF74-1. Next slide. All right, so more details on these results. By comparing these and comparing them with other gases, we saw a direct but non-linear relationship between the binding energy and the binding distance. The exception in this case is seen in ethane for the MOF74-2. Part of this may be because there's actually less distance between the oxygen atoms and the ethane molecule, even though there's more distance between the metal and the ethane molecule. Like I said earlier, there's higher binding energies in general for MOF74-2, exception with carbon dioxide in one case. The binding geometries between the two structures are very similar, with the one case I mentioned earlier. And then the gases on the top row of the top figure are consistent with what has been reported earlier. So that's a good indication that our model is working well. If you look at the bottom figure, look between B and C, that's first is the smaller structure, the MOF74-1, and the next structure, MOF74-2, you actually see that the ethane orientation has turned almost 90 degrees between the two different structures. Next slide. All right. So this is for comparing the cluster model now to the periodic boundary conditions model. Starting with the tables, we see that the angles for all the adsorbents are very similar, but the binding distances for the cluster are all about 1/10 of an angstrom closer for most absorbates, and this influences the binding energy. We see, in general, a higher binding energy for the cluster model, especially when you look at CO2 and water. And then if you look at the graph, we have all the models shown. I just want to point out that the purple color is the cluster model. It didn't make its way into the legend for some reason. Although there's no experimental data on MOF74-2 for this absorption, we see that it has a higher binding energy than MOF1, and we see that the cluster model shows inflated values over that. I don't think that those values are actually, for the cluster model, the more accurate ones, because for this case, we had a relatively small basis set and that we did not correct for the basis set superposition error or BSSE. But we have no reason to believe that these numbers are hugely wrong. Next. So some conclusions. We see that MOF74-2 absorbs gases more strongly than MOF74-1. This is actually an exception to the trend typically seen in these similar frameworks. Generally, when you have a smaller cell and with an analog with a larger cell, the smaller cell will absorb things more strongly. In MOF74, this is not the case. We see that there's effects on the binding from multiple parts, not just the ligand size. We see ligand matters where the gas absorbs in relative to the oxygen groups. We see that there's some effect from the cell expansion and contraction, and that there might be effects based upon the interactions between other gases in the cell. Then we see that our new model of the cluster can accurately predict the geometry. We have yet to see if the binding is accurate, but it also should be considered that the BSSE, basis set superposition error, was not included. So future work, first and foremost, to computationally correct for the BSSE in the cluster models, try to use a more complete basis set, maybe a correlation consistent one that's better at these long range interactions. And then we'd also like to trim the cluster so we can use even more advanced computational techniques. We use DFT. It'd be nice to be able to use MP2, something that is better at the long range interactions. And then we'd like to see if this trend continues as we continue to expand the structure. So if we had a third benzene ring between the metals, or a fourth, or if this trend continues using a different metal. So if we use zinc, nickel, or iron, would this same trend of stronger binding as the cell grows hold true? Next slide. I'd like to acknowledge Dr. Valenzano for her help and her guidance in this work, for the Department of Chemistry, and then for the use of Superior. Thank you.
Just on time. Excellent, Thomas. Great presentation. Now I would request the judges, if you have any question, please unmute yourself and ask.
Okay, I'll go first. Yeah, it was great talk. But since it's like outside my scope, maybe it was a little bit difficult to follow. So I have three basic questions. One is that can you elaborate more about kind of this computational method you use, this PBC, and you said like DFT, like for the future method consideration. Second question is, so you kind of said some material, some MOF has better absorption. So can you talk to us like why this absorption characteristic matters for any type of application of this MOFs? And third question is there any way to experimentally validate the model prediction?
Yeah, I will be able to address all three of those. So first, with the terms, I apologize, wasn't able to get all the definitions in there. So PBC, periodic boundary conditions, because this is a periodic system, that it's a crystal that repeats over and over again, it would be near impossible to have thousands and thousands of atoms modeled accurately. So PBC takes one unit cell and using, I'm using another physics term here, sorry, Bloch's theorem, it takes the effects of the cell as, trying to explain this in a simple way. the properties repeat periodically. So between one cell and the next, you'll find the same properties, same geometry, same wave function. That's what we really use to calculate this, and we'll in the details at each point. So we're able just to model one cell with repeat units, simulating how other cells would affect it. DFT is density functional theory. It is a quantum chemical technique that treats the electrons as a density and doesn't treat every electron explicitly for every part of the interactions. Saves a lot of time computationally, but is also quite accurate.
Mhm.
I think that's it for the terms that you asked about. Then you asked about what makes the separation or the stronger binding important. So
Yeah, absorption. Yeah.
The big thing is gas separation, gas storage. Take example like flue gas. Like it's a big thing to be able to capture CO2 to clean, to scrub. The more strongly something will bind a particular gas, the higher percentage of those adsorbates you'll catch in real life applications. Or if you wanted to separate two very similar organic molecules, the stronger binding energy of one of those would be helpful. It'd be allow for a bigger difference in energetics to choose one over the other. Third question was experiment. So there are experiments on the smaller one, but there have not been very many on the second one. I found one experimental study that did not directly address the binding energies, but there's been lots with a modified version of the structure already trying to overcome some of the weaknesses that it has. So none of this exact structure. And that's why I wanted to put computational study out there.
Yeah, so kind of, can you specify some application? What application this binding to specific gas matters? Because I just don't have any knowledge about those.
Yeah. So if you could use it for storing gases for like a vehicle. You need to be able to, if you're using like methane or hydrogen for a vehicle, as these applications are being looked at, you need to be able to store the gas in an efficient way. So the high volume or of the cell being able to hold lots of gases is beneficial. So that's one. Another would be separating gases. So in the fuel industry, you have a whole mix of molecules. It's beneficial to be able to absorb one more preferentially. That way you can separate out another. Maybe something that has in distillation almost zero temperature difference in what is able to be distilled. This might be able to assist that in separating those more energy efficiently without super long columns.
I see. Yeah, thank you.
Mm-hmm.
Right. Thank you so much, Thomas. Thank you, Dr. Han. I see Dr. Jacob had to leave because he is judging for another session. Do we have any question from the audience? I know we're just 1 minute past 1, but we have 2 more minutes for the audience question. So going again, do we have any more question from the audience? I'll just give audience 20 more seconds. If you wanna type it in the chat, go ahead, you can do it. Otherwise, you can unmute yourself and ask a question. All right, seeing none, I would like to thank you all for joining us, and I would like to invite you to attend our award ceremony today at from 5 to 7. And you can find the link in the GHG's website, which is ghg.mtu.edu/grc. And thank you so much for joining us. Have a great rest of your day. Bye.
Hi, Rennie. Good to meet you.
Thank you. Good to see you too.
Yeah.